Look closely at this microscopic view of biological tissue. It is rapidly stiffening into a dense, rigid, fibrous scar. Tissue fibrosis operates as an aggressive, self-reinforcing pathology that actively overtakes healthy cellular structures. This destruction is driven by a cellular-level event, the transition of dormant cells into hyperactive states. The primary engine driving this transition is the myofibroblast. It is a specialized contractile cell for wound healing that, when overactive, continuously produces rigid scar tissue. Once formed, these myofibroblasts aggressively deposit dense extracellular matrix, physically choking out normal tissue function. In clinical settings, isolated single pathway inhibitors frequently fail to stop this relentless cellular transition. Blocking one signaling route usually forces the cell to reroute its instructions through an alternative pathway. Halting fibrosis requires a systemic, multinodal intervention to prevent the initial transition of quiescent cells, rather than attempting to treat the resulting scar tissue after the fact. This diagram illustrates the transition, highlighting interleukin-11 as the crucial pathogenic driver. IL-11 is a signaling protein used to trigger structural changes. Once active, it forces surrounding cells into senescence, while simultaneously driving continuous extracellular matrix production. The Fasting-Mimicking Diet, or FMD, offers a specific dietary intervention capable of neutralizing this exact pathway. The fasting-mimicking diet functions as a highly targeted systemic signaling inhibitor. By imposing strict nutrient parameters, FMD prevents fibrosis by simultaneously dismantling the transcriptional, epigenetic, and metabolic precursors required to synthesize and empower IL-11. To understand how this intervention operates, we must first look at the microscale environment along the membrane of a single fibroblast cell. This cross-section shows a TGF-beta ligand as it binds to an external receptor, acting as the primary transcriptional trigger required for IL-11 secretion. TGF-beta is a secreted protein that controls cellular growth, while SMAD3 is an intracellular messenger protein that carries the TGF-beta signal directly into the cell's nucleus. The binding event triggers an intracellular cascade, resulting in the phosphorylation of SMAD3 proteins, which then travel downward to dock directly at the IL-11 gene. Both fibroblasts and epithelial cells heavily utilize this specific TGF-beta and SMAD3 cascade to initiate IL-11 synthesis. The fasting-mimicking diet systematically dampens this exact cascade by stripping the local environment of available TGF-beta. Without the external ligand present, the external receptors remain inactive, and the intracellular SMAD3 proteins remain stationary. By dampening this cascade, FMD cuts off the primary transcriptional trigger. This prevents IL-11 from ever being synthesized at the base cellular level. Moving the narrative focus laterally, we can observe the broader immune microenvironment surrounding these fibroblasts. Hyperactive immune cells stationed here secrete high volumes of pro-inflammatory cytokines, specifically TNF-alpha and IL-1 beta. These inflammatory cytokines work powerfully in tandem with TGF-beta to promote the production of IL-11. Chronic exposure to these cytokines activates specific epigenetic regulators inside the cell, creating a secondary, hard-coded pathway for fibrosis. Inside this conceptual nucleus, we see the JMDM3 demethylase physically unwinding and opening the promoter region of the IL-11 gene. JMDM3 is an epigenetic enzyme that modifies how tightly DNA is wound, physically exposing hidden genes so they can be read by the cell. This physically open promoter is a strict prerequisite for high-volume IL-11 expression. Transcription factors cannot bind to closed DNA. Here, we see the second FMD intervention in real time, the induction of widespread autophagic clearance, resulting in hyperactive immune cells collapsing via rapid apoptosis. This targeted cellular death significantly lowers the systemic levels of circulating inflammatory cytokines. By resolving this chronic inflammation, FMD effectively removes the upstream epigenetic stimulus, the JMDM3 activation, required to open the IL-11 promoter in the first place. Pulling back from the cellular level reveals a macro view of the organism's systemic metabolic state. Here, hyperactive insulin and IGF-1 receptor crosstalk operates as an independent metabolic driver of fibrosis. IGF-1 signaling refers to the pathway activated by insulin-like growth factor 1, a hormone that stimulates cellular growth and multiplication in response to nutrient intake. This systemic network map shows how high activity at the insulin and IGF-1 receptors funnels directly downward to prompt IL-11 expression in pathogenic tissues. The third FMD intervention addresses this by plunging the organism into a profound nutrient-deprived state. This specific state induces a systemic suppression of both the IGF-1 and insulin signaling axes. This nutrient deprivation creates a strict metabolic blockade, cutting off the signal from the top down and preventing the systemic expression of IL-11 before it can begin. Even with these blockades in place, we must account for scenarios where trace amounts of IL-11 still manage to secrete into the tissue microenvironment. Secreted IL-11 alone cannot build scar tissue. It must activate specific downstream metabolic effectors to actually drive fibrosis. This diagram outlines that exact network of downstream effectors activated by IL-11, the ERK, mTOR, and JAK-STAT3 pathways. The JAK-STAT3 pathway is an internal communication chain that relays chemical signals from outside the cell membrane straight to the DNA promoters. Activating this specific three-pronged network is what finalizes cellular senescence and instructs the cell to begin extracellular matrix deposition. FMD imposes a secondary metabolic effect. Due to the nutrient deprivation, the body maintains strict systemic mTOR inhibition. This strict inhibition creates hard cellular resistance, physically preventing IL-11 from executing its instructions through the metabolic pathways. Because the downstream pathways are blocked, FMD acts as a comprehensive failsafe. It short-circuits IL-11's pro-fibrotic signaling, even if the initial expression blockades are bypassed. This master circuit board combines all three pathways into a single view of the IL-11 fibrosis machine. First, the blockade suppresses the TGF-beta transcriptional trigger. Second, it resolves epigenetic inflammatory stimuli via autophagic clearance. Third, it locks in strict IGF-1 and mTOR metabolic resistance. This additive, multimodal inhibition provides a distinct mechanical advantage over isolated therapeutics, which leave alternative pathways open. Here, we see the ultimate physical result of this microenvironmental blockade. The dense, rigid extracellular matrix dissolves, revealing healthy, quiescent tissue. This multinodal intervention starves the fibrotic process simultaneously at the epigenetic, transcriptional, and metabolic levels.